Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Today we have Klaus Lundegard with us to give us an interesting talk about immunology and computer science and the intersection between the two. Klaus has received his PhD in protein chemistry in '98, and since then he has worked for small computational biology companies or bioinformatic companies, right, for a couple of years in San Diego. And since 2002 he's been a professor at CBS at Technical University of Denmark. And today he'll talk about immunology.
It's not full professor though. It's a lecturer though. Okay, so it's.
Oh, you better go.
So yes, thank you for inviting me. I'm very happy to be here. Interesting talking to you all afterwards also. It was good that I met you guys down there in Israel. And also in Budapest, actually. So first, I'll give a brief introduction to the center where I am. It's all headed at the Technical University in Denmark. And it's this CBS is formed in 1993. We had got a huge, in Danish standards, it was quite a big grant. It was 2 times, it was this 10-year, which was called by 2 times $1 million grant that made CERN available to set the group up and start these things out. That has turned out to be really good. The background, even CERN is a physicist really, and then started in something like a sentence recognition and started out with machine learning. He had focused on having it bio-based. So most of us has a bio background that then uses the algorithms that other people develop in different biological problems. So that's what we do at CBS mostly, and then we also do some teaching, and even some of the courses are internet transmitted, so you can follow them even though you are abroad. It won't really work from here, I guess, because then it's the middle of the night. So, but if you are a person that are awake in the night, then you could do it. And also we have a huge website with prediction servers. It's also something that CERN has focused on, that all the things that we develop should be available for the people that would like to use it. And actually nowadays most of the funding is coming from EU and NIH and not very much from the Danish government. So, and CBS has these 8 research groups in very different fields, all the way from, we have nutritional immunology, which is not really at all computer based. They have just been fused to the CBS group recently for strategic reasons, but they have then lab facilities and knowledge in immunology that we could very much benefit from. Yeah, they're working with how, for example, the bacteria that comes from the food and the gut flora, and how it interacts with the immune system, and so it's something like that, but also allergy and things like that. Also have chemoinformatics groups doing something working on small molecules. But basically, the main focus area is on what comes out of sequence analysis, and then now also turning more and more into integrative systems biology, and also the field of DNA chips and array studies, also combine that with systems biology. Yeah, I think I'll just skip. This is just And then some commercial here that they actually, all these collaborations actually have turned out to some very, very strong papers during the time. So, and this is our website where we can go in, and there's the two major points is of course the prediction servers, and then there's the courses. We have this huge diversity of courses.
Anyone can watch the course, whoever likes.
No, you have to apply for it.
Okay.
To go and join the course. That's the university rule that they have to. I mean, and if you're outside the EU, you even have to pay for it and stuff. So that's. So, and this is how the prediction servers look. Actually, we are redesigning this page because it's go quite a lot further down here. So you can't see everything here. So these headings might be put into top, and then you can go to the fields that interest you. So that was for CBS in general. So I'm from the immunological bioinformatics group that is headed by Ole Lund, group leader. We are 2 associate professors and a number of postdocs and PhD students right now. And we also have a number of collaborators, very strong one with the Copenhagen University, which is very close by and it's very convenient, and Søren Buch's group where they're doing biochemical assays of the predictions that we are making, and also Mogens Claesson's group, which are doing the ELISpot assays of the predicted epitopes. But otherwise we also have for example in Tom Ottenhof's group in Leiden also did some ELISpot assays of particular tuberculosis epitopes, and we also have the collaboration with Alessandro Zet and Bjørn Peters on the epitope database IDB. So our funding is basically also EU and NIH. So we were so lucky that we actually together with Søren Buch's we got an epitope discovery project from contract from NIH, and also so one is Søren Buch's PI and the other one is Ole Lund's PI. But it's more or less half the work on our place and half the work on their place in both cases. So, and this is what we are doing, selection of epitopes and trying to find reactive peptides in TB for also for diagnostics, and cross-reactivity prediction also we try to do something, and then we are also working into the systems biology by having 2 PhDs working in simulating the immune systems, with trying to integrating the different predictors that we have made, that I will talk about some of them here, into some system that could simulate what will happen with in the first case with the pathogens, and for example the strong evolving pathogens like HIV, what will happen when they come under the pressure of the immune system.
Are you going to say anything about predicting cross-reactivity?
No, not in this I have here.
Can you say a few words about it? What inputs do you use to predict cross-reactivity?
Yeah, it's what we're doing is actually I can say for the T cells it's the negative cross-reactivity we're trying to say. So we have made this see how the distance from a peptide to something in the human genome or human proteome makes it less likely that it actually is an epitope when you go and look at the pathogens. Yeah. So that's so just briefly go over the immune system. I guess might know some of that, but so the vaccination is just you put something in from a pathogen into to a person, so we will react against the pathogen when we see it in real life and not getting killed by it. And traditionally it has been made by killed or not so reactive types of the pathogen. And it works like this, where you have first time you get a weak response, but the next time you meet the same pathogen having this antigen, you will have a stronger response. And this has been highly effective, especially with pox, that was where we have the World Society have managed to actually eradicate the disease in total. This might be some lucky event, because I mean this was not highly epidemic at the time where it's not like because something like measles or something where we're also vaccinating and there's something that is around all the time. I think it would be hard to actually eradicate it totally, even though you have an effective vaccine. But still, it's a highly effective way of avoiding getting sick. So the immune system basically consists of these two legs: the innate immune system, which is the thing we are born with, and the adaptive immune system, which evolves while we live. So the innate immune system, it's unspecific and basically antigen independent, but it has an immediate response that would turn down an infection so much that hopefully the training of the adaptive immune system will come in and remove the things. And there's no memory in this. And the adaptive immune system, on the other hand, is pathogen specific. It depends on which kind of pathogen it is, how it will react. And there's also two legs, where they have the humoral and cellular part. So, and it works with, you have a pathogen, and in the pathogens there will be antigens, which are the substances or proteins that actually the immune system will react against. And the parts of the antigens that the immune system reacts against is called epitopes. That might be something structural, like which are the B-cell epitopes and antibodies, or it might be linear parts. Could also be in this working with antibodies, but also for T-cell epitopes. So, and why this work is because the organism, human organisms, have a very high number of different receptors for the epitopes. So basically, except for the cases where there will be this negative cross reaction, there will be something, a receptor that would react to anything, more or less strong. And then there will be a second selection will make the binding stronger to these parts. Humoral immunity, which is the antibodies, is working here. You have an antigen. This is the so-called Fab fragment from the antibody. Yeah. There will be two on an antibody, and that can be in soluble form or as a receptor, B-cell receptor sitting on the surface of a cell. And then we have, on the antigen, we have the epitope part, which is the part that binds to the antibody. And the parts on the antibody that binds to the epitope is called the paratope. So, for this, again, we have here the prediction servers, and then when you scroll far down, we come to the immunological features here. And for this, for the linear parts of the antibody epitope, the linear B-cell epitopes, we have this called babypred, which is a predictor, which has been, I won't go so much into that. That is not something we are developing on right now. And then there's the more interesting parts, which is the discontinuous epitopes, which has been developed by, actually it's basically what's standing here on the top here. They have developed a propensity score, see what kind of amino acids is actually in epitopes. So we're making a propensity score for amino acids for how likely it is that they are in epitopes, in B-cell epitopes. and that combined with the contact number. So in this case, you need actually the structure of the protein. So the contact number is how many atoms is in the neighborhood of a particular atom.
That's like C beta score?
Yeah, but it's
Like the number of carbon atoms.
yeah, it's and I must admit I can't remember exactly if it's the all atom calculation or if it's only on backbone backbone atoms.
The propensity score, how do you compute that? Like the propensity for each amino acid is what?
Yeah, that has been. I mean, you have this selecting. The data set has been taking epitopes from the PDB. That is our training and evaluation set. That is what we're defining as that's real epitopes. You have an antibody antigen complex with a structure in PDB, so we can see which parts are actually in contact. And the part from the antigen that are in contact with the antibody, that is what that part. That is the epitope.
Okay.
So, and then we look at these epitopes and the amino acids that are in contact with the antibodies, and then we statistically analyze how often do these occur, and also, and of course, we're doing a background normalization to how often do they occur in nature in general and stuff. So
it's just a measure of frequency normalized by frequency in nature.
exactly, yeah.
And is that what comes out like? Is it very similar to the frequency in nature, or is it quite different from it?
No, it's for some. I mean, some things are not very surprising, but very different from things that, for example, tyrosines are very often located in this kind of interactions, so there is some difference, and for some amino acids there's a significant difference, and that's why it works. Really, it's you can.
There's a significant difference between their frequency in the population and the frequency in
epitopes. Yes, exactly.
Is there somewhere where we can read off this propensity score directly?
I think they are published. Either they're published in the paper, or there will be there's extra material. Extra material.
Where was this published? Do you remember? Oh, it's down there. Protein Science.
Yeah. So I probably also have the paper on here somewhere. I can fix it. So, and then I'll go to the cellular immunity, which is then when you have all proteins that are expressed in the cell will be degraded by a complex called the proteasome. Some of the peptides that are coming out of this degradation will be bound by a transporter complex called TAP, transporter associated with antigen presentation. And the peptides that are then coming into the endoplasmatic reticulum, some of these peptides is binding to the MHC, the major histocompatibility complex class 1 that are expressed within the ER, and then they are transported to the surface and presented on the surface on the cell. And then CD8 T cells, if they have the right receptor, they will bind to the MHC with the peptide. And that if this part here is something that and if it has the receptor, it means that it has not is something that we haven't seen before, because otherwise it has been the receptors are deleted in the thymus during learning. So and that means then that's a signal that it's something that it reacts to, the signal that the cell should be killed because there's something wrong with it in some way. So, and this is how it looks in real life when such a cell got. This is the T cell, and this was the infected cell, an influenza infected cell. So, but starting with the proteasome complex here. Oops, wrong way. Yeah, this is what it really looks like, and it exists in two forms, where the protease reactive parts can be exchanged, and that is with it's induced by interferon gamma, which is a signal that something is going on that the immune system should react to.
So the left side is just the proteasome
that constitutive proteasome, you call it. That's the ordinary proteasome, which is going on all the time. And then if something happens, these subunits are exchanged to this more specific subunit. I'll show you in a minute. So, and this is just how they evolve. But see, the constitutive, there's a human. The human is the HS, yeah. So this is very close to actually to both mouse and rat. And there hasn't been so much involvement in the proteasome is actually pretty conserved, especially the constitutive part. The constitutive, can't see that here, but it's not actually not very different from the yeast one, how it functions either, the constitutive proteasome. The immunoproteasome is a little bit more specialized, and you can see that in the humans are clustering with the bovine.
So if immunoproteasome chops things differently than proteasome, then early on when you delete the receptors that recognize cell, somehow this immunoproteasome has to be turned on for the body to learn. Otherwise it might attack anything.
As I'll come a little bit back to it, but for now I can say that it's more like the constitutive proteasome is more stochastic, so that makes everything. So and the immunoproteasome is more focused on the thing that the immune system can actually react to. So even though it's only the constitutive proteasome going on, it will still make the peptides that the immunoproteasome is also making. So it's just then when the immunoproteasome is turned on, it'll make more of the relevant one, and that's important than when the immune system is triggered.
What's the advantage to having constitutive at all?
I mean, the basic, I think the constitutive proteasome, or just not having the immunoproteasome on all the time.
Yeah, it doesn't work all the time.
Why?
No, yeah. Why? Maybe.
Other functions in the cell.
Yeah, maybe because it's making general smaller peptides, so it's that probably more of the amino acid will be recycled the correct way if it's not turned on. Otherwise, more of the peptides would go into the ER and have to go out again, and that's probably not good for energy. I don't know for sure, but that could be a guess.
But it's constantly degrading proteins of the cell.
It's
its primary function. So.
Yeah, the primary function is really recycling, and that's it. Just seems that I have cut that out of this presentation, but it just seems that actually then the other part of the immune system, the TAP and the MHC, has learned to focus on some of the peptides that the proteasome actually are making, and making benefits of those that were already there in the beginning.
So another question is, how do you? First of all, this looks like highly non-specific.
Yeah.
Especially like even the immunoproteasome won't be able to chop up everywhere.
Yeah, it's more or less. Yeah.
And then the other. So it's a very mild signal. But the other question is, how do you measure this, and how do you separate it from MHC binding?
Yes, that depends on how you do it. Yes, there's a way to. There's a number of different. There's probably more than this, but this.
What sort of data? How do you measure it in biology?
That'll actually. It's just so. Let me actually start here. So this is one way to do it, that you find epitopes and find out, what is the context they're in the real protein, find the real protein. So this is, and then find that because there's so far nobody has seen anything else that should make us the C-terminal other than the proteasome. So the proteasome is, at least for the vast majority part, responsible for the C-terminals generated. So this is the cleavage side of the proteasome, and then you can extend it to the protein that the epitope was coming from.
But there should be some generic correlations between neighboring amino acids anyhow, right? I mean, if you see...
Oh yeah, of course you have to take into consideration what is the statistic of the general case of how does it look when you have a valine here. As soon as you go into dipeptides, it's not, the signal is almost gone because there are so many. But of course there is a bias in when you have a valine. What is the neighboring amino acid there?
So you take that into account when you're learning NAT shop. So you look at the general statistics, all comes up to valine.
Yeah, because it does. We are using neural networks to train it. So it's inherently, because they also have negative, and for the negatives we are using the parts inside. But actually we are doing first a training round where we train it and using all of the parts inside as negative results, and then we are saying, okay, maybe we are then predicting on everything and see, okay, the ones that are very high predicted as cleavage site inside, we won't train us them as negative results, because there might be a chance that they are actually positive as because it's stochastic, so they might also be cleaved inside still.
And when you're learning the chopping, are you taking into account your epitope predictors as well?
Not for this. No, this is for the net chop is only.
I guess your negative example should be something that has a very low binding energy. It's highly likely to be epitope when you look at the 9-mer, but it isn't an epitope. It is not a known epitope. For example, in HIV. So then you look at the boundaries to see what are the boundaries.
Yeah, but it could also be for other reasons, like the T cell receptor. But there is a strong signal also here. I mean, if you're just taking this, you can actually use this to predict epitopes with, because it is inherited that this part here, it looks like also something that binds to MHC because it comes from MHC. So it's biased in that way. So
how do you define epitope in these studies?
Okay, maybe I should just say peptides, really, because it's peptides that is taken from Suffiety database. So it's eluded peptides. It's peptides that are presented on the surface of cells, but not necessarily are epitopes. Really.
Do you guys have a standard nomenclature to make these distinctions? Now we keep struggling.
Yeah, but
Epitope is a different than another person's body.
Yeah, but you should say, I mean, in this case, I guess you should say eluded peptides. Or
is there a standard nomenclature emerging?
I don't know, but actually maybe you should just use what, I mean, the Suffiety database has been there for a long time, and they're using, they're called these natural ligands.
Natural ligands. Binders.
No, because binders can be something you put in outside, so then they might not be processed.
Ligands. I see, they come from the inside.
Yeah.
Okay.
So it's, yeah. So, yeah, but it's.
And when you hear the word epitope,
do you think, full T cell receptor, gamma interferon release?
I've started to do that because we actually now, we also, as I will show, we are integrating the whole thing into the real epitope, the epitope prediction. So the epitope is the one that actually triggers the immune system. These are peptides that are being processed and binding to the MHC and presented on the surface.
One other question. Proteasome, you think of that as a set of things or one thing? Set of molecules that are doing?
I mean, we're not treating it as a set of molecules. We're treating it as two. I mean, there's the immunoproteasome and the constitutive, and that's it. I mean, we're not departing it into the different subunits that are actually in there. You could.
But there could be different sets of molecules that make up the immunoproteasome.
No.
And all have different specificities, and we just don't know because we're measuring it at a gross level.
I'm not sure. I think that it's either or. I mean, it's either the one set of subunits or the other set of subunits together. But each of the I would say, is it 6 or 3? I can't remember. Okay, it's three different. There's 3 different subunits, and each of them doing a little bit different, have a little bit different specificity. That has been shown, but we are not going into details about which shopping.
And do those mix and match in nature, or?
I don't think so. I think that you either have the betas, this from the constitutive, or the ones together from the immune. But I won't bet on it.
So have people sequence that area of the genome and look for differences, individual differences, and haven't found them, or?
Yeah, because they've done the phylogenetic testing, so they must have sequenced.
Yeah.
Well, but they've done it across organisms, right? Have they done it multiple times in different people?
Oh no, but there's no. As I said, there's basically no divergence between very non-related species. So in between humans, it's basically all the same. And there might be, of course, some point mutations in some, but it's nothing that seems to be important. At least there might be diseases that it could be important for, of course. And then the other.
When you said that it's highly conserved, you really think highly conserved. You're thinking conserved at the level of like MHCs, for example, are also
they are
conserved.
highly diverge.
If you look at across the species, you can actually track probably the evolution of MHCs, so you can see that that all the animals had MHCs.
Yeah.
But
within the molecules they are very diverse.
But here you're saying that between us and yeast, there's virtually no difference.
There's very little difference between even between us and in all eukaryotes. There's not much difference, and especially when in all mammals there's basically very little difference. Except actually that's not in rats. You see that there actually is some population difference. So in rats, there seems to be something going on within the proteasome, also with the diversity. So far, I mean, There might be, of course, other animals as well. So it's not. I mean, but in general, it's not. And in human population, I think you have not seen anything that would say that it's in general.
And I haven't found diversity.
People have checked, yeah. So this is then the other way. This is then taken from the constitutive proteasome, and it's been done in vitro. So you put in peptides, or like here a whole protein, and make the proteasome digested in a test tube, and then you find out the peptides by HPLC and mass spec to identify the different peptides and also the amount of peptides. So you can say to some certainty the frequency of how often is a special site cleaved and not. So in this case, you get actually
So it's real biologically measured shocking data.
it's biochemical measured, yeah. So you are getting a real number value for each site that's somewhere between 0 and then the ones that are the most cleaved, like this one is very often cleaved. But as you see, I mean, many sites are cleaved, and actually if you do this again, you wouldn't get the exact same map because there's
A lot of noise.
there's a lot of noise, yes. But in this,
Lots of times.
Do it lots of times, take the data and train.
yeah, but it's really, I mean, we are working with people, it's expensive and very hard time. It's very hard to get the proteasome do it at all, especially on whole proteins. On peptides it's a little bit easier, but it's they have major problems experimentally.
So how much data is available?
Not very much. I mean, I think for whole proteins there's I think there's maybe 4 or something in literature, fully digested proteins. And one of them is for the Scrappy agent, and that's not good for anything. It's behaving strange, comparable to other. So there's maybe 3 proteins you can use. So then we are measuring the performance you can do with the, like standard and Matthews correlation coefficient. Just you don't need this. It's just, or the A-rock curve like this. Rock area under the rock curve. So, and in this case, so I think this one should really be switched. I didn't come back to Morten. It's Morten Nielsen that made this slide for me to actually figure with him, because in this case we can get a Pearson correlation coefficient, so it means that it probably is the real value data I have here. And for this, where we only have the Matthews correlation coefficient, it'll be the on-off the ligand non-ligand
Can you
thing.
get real value data off of it cleaves or it doesn't?
I mean, it's real, but it's a real number value, I would say. I mean,
What's the continuous measure of? It either cleaves or it doesn't.
no, for the it cleaves more than, I mean, it's how the probability of the cleavage. It's so. And this is just but also to show that you can have a very high sensitivity in sometimes, but it's always on the cost of the specificity, of course, and I mean, a lot of times when you see published paper, people doesn't take this into account, and they only publish one of the values. Yeah. So, and we have a server for this where you can actually, I can see, but you can choose here which one you want, if you want the one that is trained on the C terminals or the one that are trained on the. And if you're using it strictly for epitopes, so because of this inherent thing that the ones used for MSC, so this one is of course better, but you wouldn't use proteasome cleavage as the only thing for epitopes anyway. So it's I can say that now. Anyway, when you go back to if you combine it, it doesn't matter if you're using the one or the other set of data. They're giving equally much into it. So you are getting the same signal using the C terminal cleavage sites as using the biochemically determined when you combine it into the whole thing. It's just that you get some more when you are using the ligands for training. The TAP predictions, we are also working on that, but actually, and for the binding to the TAP, we have something that may be slightly better than the one that Björn Peters has made. But when you integrate it, Peters' approach seems to be better. So we are using his approach with this matrices, special matrices, where you can also use negative data to train.
Do you think it's the data that he's using to train that is making?
No, I don't think. I think it's. I mean, this matrix method is really good. I mean, yes, exactly. No, it's not. It's not taking higher correlations into account. Yeah. Yeah.
Regular lines. Has anyone tried fancier nonlinear methods?
We have. That's what we did. So
it doesn't work as well as the linear method.
It works just as well as the linear when you integrate it. But if you're looking only on the binding, the machine learning approaches are better. You're only looking for the binding on the TAP, but when you integrate it into find out what comes out in the end, then it dilutes out and they are equally good. So
yeah, I'm not finding.
This is, for example, with an out of sample set like HIV. Where you test on
exactly. Real epitope.
And then it all washes out.
Then you.
Finding the same thing.
Yeah.
It's probably.
I mean, there's other things that are going on too, the T cell receptor and all that.
No.
But no, it's not. I mean, we can get an improvement as I've shown to include all things, but it just doesn't matter how this and this is. I mean, these two this is very stochastic, and the tab are basically binds only what can bind to some MHC in some extended version sometimes. But it's so there is not very much signals in these two levels. Yeah. So this is the tab, how it looks, and this is then how the low, if you're looking only on the 9-mer, and you see it's very, I mean, the signal is not very high, basically, anyway, for the ends, but it's in the ends, the 3 first parts and the C terminal that has all the signals. There's no signal within.
And I take it there's, like the other case, there's a relatively small amount of data where biologists painfully measure tap binding.
Yes, we have a collaboration in the EU project with the Fernandez. So there and we have some iterative process. So now we are predicting with the neural network trained things to try to get some especially good peptides for training. I mean the ones that the predictor is not very sure about, to see if that helps. We had some indication that could actually help in some systems, that if you train a number of predictors on slightly different ways, and then you look to see how they predict special peptides. If there's big difference in the prediction for this between, we might think that there might be using that if we have that data, it might improve the prediction more than just using some random peptide of the data. Yeah. So, and that we have a small EU project for investigating if this is really true, and there we can send to Peter Fernandez. He then measure some number of peptides. So that's very nice. That's in progress, yeah. Because again, there isn't in the literature, there isn't that many data to get at all. I don't know if they are going to be a part of the IDB also, this kind of data.
Sure.
It would be a good thing, I think, anyway. So, and there you see that actually it's basically the first tree and the C terminal that's making the. So, yeah. And then comes to the MHC binding. So it's class 2 with a peptide in. See the anchor positions is in the end where the deep pockets are. So, and different alleles has different anchor positions clearly if you're looking at a high number of peptides. Some are more alike, and then you group them into the supertype concept. But then again, I mean, they are alike, but they are not identical. You see the position 2 here between the A6802 and the A0201. There's clearly a difference, even though there's a lot of peptides that will bind to both of them. So yeah, this is just how I've been involved, like just looking at the anchor position, and then have some more extended motifs that they're doing is Phyte. Then, yeah, this is, I should have corrected this. This is some in our terminology, because we started out doing real hidden marker models to try to, but we ended up that it was only PSSMs in the end, because there wasn't any transition states. So it's really so we shouldn't use marker model as the name here. I try to change it.
What do you do for 8, 9, 10, 11 mers? How do you just train them separately?
I will. No. I can do. Let's just. I can go. Yeah. Okay. No, maybe I should.
If you have slides, just wait.
I have slides. So. Yeah. Okay. So we have here. We're using. This is just the ordinary information matrix where we have just the statistics and the information content here. And the did I say that here? Yeah, the information content and the entropy. Yeah, and that's what really is shown here in the logos as also as I showed before. But then we are using. I mean, this is just if you have a small number of data and you're just using information. Content, you're not getting very clear signals, so you can do this sequence weighting and pseudo counts. And what we are doing is actually basic like the Altschul thing we're doing in SciBlast. It's exactly the same, and using Blossom 62 as the background for calculating the pseudo counts. And then we are using a weight of 200 on the pseudo counts. So if we have 200 real data, then half of it would be consisted by the pseudo counts and so on. That has an iterative process to see what would be the best, but that's what we see.
The clustering just clusters together
Similar peptides.
No, there's no clustering here. It's just to show that this would be downweighted if you have equal sequence. Yeah. Okay. So in this case, there's. The clustering goes to downweight sequences that are too alike, to avoid that you are getting a high signal from one position that is not really true. And they're doing the same also in the SciBlast when they're. So so we end up with this position-specific scoring matrix with the. In this case, we have the probabilities, but we're using the log-odd ratio, so we can just sum up over the matrix and the positions to get the score from so like this. So this is for the first peptide here, right? So sum up and you get a score here for these three peptides, and it actually fits pretty good. It's working very well with the matrix method thing.
So it correlates well with the actual binding prediction?
Actual binding prediction. So we have here, and this is what you see. We have here the very simple, and then if you go up to use both sequence weighting and pseudo counts, and if you then use the knowledge from other, for example for sulfite, if you know which the anchor position are, you can also upweight those. We're doing that if we have to use matrices and we don't have more than less than 10 data points, and there is some information in sulfite about the anchor position. You can use that and actually get a decent performance.
What do you mean get information about the anchor position? What does that mean?
That means that they have, for example, if they have done peptide libraries scanning, then they have seen that these two positions within the anchor positions are not. Usually they are 2 and 9, but not always. Like in A1, that is 2 and 3 and 9. There's 2 anchor positions, and they might. But if you know what the anchor positions are, you can
allele.
For that particular allele, then you can upweight these positions and get a better.
But you are not using the
same data to train, or
No. That's then because you're not using the peptide library data. You cannot train on these. These are not, or they might not be available anyway. So the ones that you train on is the eluded binders, but there might not be more than this 5 or 10, and then they have this information from peptide libraries that indicates that the anchor positions are on these positions. They got
those motifs.
those are active somewhere.
But not from this.
You don't have access to that.
No. Yeah. But then the neural networks and the support vector machines, they can then take into account the higher order correlations. So you see that there might be some competition for space somewhere here. So it does matter what there is in one position to take into what can be on the next position. And the matrices cannot take this into account. So this is just like an XOR function. And this is then, if you have here, you see 313 binding peptides and just random peptides, you see that there actually are some signals that neighboring position from Swiss prot, just natural peptides, but just randomly selected. So there is a signal in neighboring on some positions. So it would be, there's a reason to do the neural networks and support vector machines. And then there's the question of the sequence encoding when you want to put them into the training.
So what is Blossom encoding?
Yeah, I hope I have. Yeah, okay. Yeah, I have there. So we can put in. You can use either this one called Sparse Encoding, where you're having for each amino acid you have 20 positions, and one of them is 1 and the rest are zeros, depending on which amino acid it is. And these will be. I mean, they are all equally unrelated. So we don't get any. The networks are not getting any relationships between the amino acids. They're just getting that these are 2 different amino acids and have to learn for each amino acid. And it have to have examples for each amino acid to get to be able to learn them, because there is nothing on the other positions. On the other hand, if you're using the Blossom matrix, this is a Blossom scores for these two. The Blossom scores from a valine and the Blossom scores from a leucine.
You just take the row in the Blossom matrix?
Yeah, you just think. I mean, on top of here would then say that would be one amino acid. The likelihood of having whatever an alanine here or whatever it is. I don't have them on top. So we have these 20 numbers for each amino acid. That is the substitution score for each amino acid. And then a valine and a leucine have very similar substitution scores. So you see, then you actually already give the neural network that the valine and the leucine are highly related. Chemical amino acids and their chemical physical properties are very much alike.
So do you encode only one way, or because you could also encode 2 ways, right?
I mean, you could concatenate D from the first to the D from the second and have a 4-dimensional feature vector, which would allow your neural net to ignore maybe the Blossom encoding if it needs to.
Yeah, but then you have a high number of inputs and you would have high number of weights. So that's the other problem. Already when you have just a 9-mer and you have 9 times 20 inputs, you have this 180 inputs. You need, you need at least 100 data points, but preferably several hundreds of data points to be able to adjust all these weights in the network properly. So so having both of them would then, because you have the hidden layer, so there will be an exponential effect of having even, or not an exponential effect, but twice as much.
You could have sparse controls and so on to make sure that all the...
But actually what we do, what we then do is actually we are using both. And also, so we are training both with the Blossom. We are training 10, actually we are training more, but we are using 5 networks trained with Blossom matrices input, and 5 of them used with Sparse, and making an average of these 10 predictions. And that seems to be better, to have the average score. Oh, it is better. It's
What's the architecture of the network? Is it a couple of layers or?
Yeah, it's 3 layer, right? Input and output, and then 1 hidden layer. And we have, and also there we have different architectures. So we have some. We even have the linear one where there's nothing in the hidden layer, and then up to 64 hidden neurons. And actually we are then selecting the ones that we are training on 4/5 of the data set and stopping on 1/5. And then we are selecting, these 10 are selected out of I can't remember how many we actually train with different architectures and different inputs. So we're taking the 10 out which has the highest, the best stop performance, and then we average over these.
I'm sorry, I missed the Blossom encoding.
How does it come? You've got your peptide. How does the Blossom matrix come?
But you're just using the Blossom matrix instead of. You're just having you're taking. I don't. You should have had the matrix itself here, but you
start with a 9.
Yeah, you're going with the 9. But this, like here, we just have a dipeptide, but you could have a 9 here. And so you go in and see for the valine. Then you go into the Blossom matrix. You have the full substitution row for that amino acid.
Each peptide, blow it out to...
To 180. Yeah. Exactly. And I mean, in general, the BLOSSOM scores are a little bit better, at least for the linear correlation coefficient. You get when you're using the area on the rock curve, so you're getting basically the same. And you see when you're having the ensemble, you are gaining a little bit again.
The DSM does so well, given that you have this impressive slide showing there are lots of correlations among the...
No, there was some correlation. I mean, okay, if you go back and see the numbers, they're not very high. So they are. I mean, okay. So it's. I don't know if actually the score was here. How to calculate the score? No, I think I have left that out. But it's not. I mean, the numbers doesn't mean it's not a striking effect. It's a slight effect. There is an effect, and you can gain a little bit from it. But it's. I mean. And that's why the
non-linear method works slightly better.
The non-linear method works slightly better than the linear method, but the linear method are working. If you have enough data, they're working quite good, basically. I mean, Sette and Bjørn Peters has used this. What is the name? Stabilized method for predicting linear. And then they did a comparison with our method, a blind thing where they actually use different peptides and go in and train our method and their method with the same data. And they saw that our method was slightly better, but only slightly better than theirs was.
That was the cross-validation data that they split into 5 splits in the PLOS compiled paper.
Yeah. So that's working. I mean, this stabilization matrix method is quite good in itself also. But actually, I didn't tell. Actually, we also use to make and that's basically maybe the network train faster, but also helps a little bit. Is actually to. Do I have this here? No, I don't show that. But we are using also. We have this 180. Inputs which come from peptide, but we're also using the 9 inputs that comes from the matrix. So we're making a PSSM from peptides in Cepheid, and using the output of that PSSM as an input to the neural network as well. That will be then will trained on different peptides, because there's not the ones in Cepheid that we have actually got measurements on. So, but in that case we are getting, and that gives a little bit also extra. But in that, the Peters and Sette, we didn't, because I mean, this concept couldn't handle that. So they was actually just making a matrix from the same peptides as was the training set for that part of the cross-validation. So each time in each cross-validation they were making a new matrix and using input for that. And actually there you don't gain anything from having this input, which is not surprising since it's redundant data. You're getting the same thing you're getting twice. So we have just made an update from the methods on the web, and also including in this update is we have some animals also, and we have made neural networks on 8 mares from a few of these. For the 10 and 11 mares, we have from 10 mares we also could get some, there was data enough to get some neural networks, but then find out that we could actually making kind of pseudopeptides. If you can make 6 pseudopeptides out of a 10 mare, where you're taking, where you're always keeping the tree first and the last, like in the tab thing, and then make the other just shifting, making a sliding window that will cut out 1 or 2, depending if it's a 10 mare or an 11 mare. So making these 6 peptides, and using the average of the 9 mare predictions of these 6 peptides as a score. And then we had 3 or 4 networks where we actually had trained on the 10 mares. Then so we could compare what would happen if we are doing.
Go back? I didn't quite catch the last slide. What were you doing?
Excuse me?
Can you just go back a slide? I didn't quite catch that. You're doing.
Yeah. See here we have the 10 mare.
Get some
fixed removal size?
So here we have a 10 mer. So we are keeping. So we are keep. That's a 10 mer, and then we're getting 6 9-mers out of that. And the red ones are always kept. So we are just only deleting.
You're saying the first 3 and the last we necessarily want, and then we slide through all the other ones, removing one at a time.
Yeah, if it's a 10 mer. If it's an 11 mer, we remove 2 at a time. But
Because usually when you have
Two at
a...
a time, 2 consecutive ones?
yeah,
Or
yeah,
two different
yeah.
ones?
Just 2 consecutive ones. We're not using all.
That if a 10-mer would bind,
You usually have something that sticks out in the middle, so that would be bound on the sides,
Yeah.
the anchor positions, and then somewhere in the middle would stick out of the binding.
But you don't know if that's a flexible situation, if it's actually moving all the time. And it is, maybe it does, because it's actually in the average affinity that is coming closest to the real
maybe
one.
it's like...
Binding to this position and in that position.
Yeah, actually it can go up and down in the middle, because it's not.
And you take the average.
And then it's taking the average, and the average in the log space it is really.
And why did you decide to fix the first 3 specifically?
Because you see that the anchor positions are usually on these 4 positions, not on the first, but the first 2, 3, and 9 are usually anchor positions. So that's and so these are important.
Based on that, okay.
Yeah.
Do you do the weird one, the BO8 separately or something? The one that has the anchor on position 3 or whatever? I'm sorry.
Yeah, we should actually we haven't evaluated this because we didn't have 10 mers on the BO8. So that is a good. We should talk with Boost to make some 10 mers on the BO8 to see how well it works on that really actually. Yeah, you see. So this is the average of the 6 log scores using the 9-mer predictions, and this is the prediction using the 10-mer networks. So on the web server, if the 10-mer networks exist, it uses that. But if it doesn't exist, it'll use the approximation. But you see here, it predicts 344 nanomolar, and in this case, it's 211. So it's pretty close. And during an average overall, you see here, it's more using the Pearson correlation coefficient. It's almost the same, yeah. But that also has something to do with we have less 10-mer data. So these networks are not that good. And the 9-mer is usually really good. So it's also, I mean, if we are getting more 10-mer data, this one would grow, I'm sure.
Yeah. So red is train and test on 10.
Yeah.
Have you tried taking the minimum?
On the 9-mer data? Instead of averaging?
Yeah, we have both tried to take the minimum or the maximum or some weighted average. The average seems to be the best. So and then it comes to the integration. So we have these 3 steps here. So we integrate them into predicting how well do we identify CTL epitopes really, and that's like. What we have used as evaluation set is like HIV, where you have a high number, I mean where you have a high probability to see if something is an epitope, there's a good chance that it has been seen, because that has really been investigated, the T-cell epitopes from this. This is the server. So, and using this evaluation set that's from Los Alamos, we've taken from, and of course we have not trained on that one. We seem to be slightly better than MSC pathway and somewhat better than what else have been.
Yeah, I talked to Morton about this.
Okay.
And because we're trying to see how well we do as a comparison, and we just can't, I think, what is it, version 1.2, whatever your latest version is. And he said, no, swear to God, we did not train on these HIV epitopes on the existing thing that's on the server right now. And we can't get anywhere close to that when we play the game the same way, when we don't train on those epitopes. So are you sure you don't have an intern there somewhere that...
Yeah, that's Mette that has done it. Yes. But are you using the same? Are you using...
If you didn't train, wouldn't
you want to train? You have all this extra HIV LAML data. Wouldn't you want to add that into your training?
Maybe that's a predictor anyway.
Yeah, but we're using the Suffici data, and there we have this collaboration with Stefan Stefanovic in the Suffici. So we are also getting a lot of Suffici data all the time. So we're using.
I think it's the extra.
So we're using this. It might be that we have the extra amount to train on. Yeah, the amount in the training set. But I guess there should be. Are these available also, or maybe not? Okay, we're trying to get this paper published right now. So and there's supplementary material. There will be all the data sets. So I guess, but maybe not the training data, because they might be prepared. They want to publish them first before they.
Yeah, we think there's a misalignment between the data that, I mean, what we have from you or from your website is here's the predictions.
Yeah.
And they're just so good that we just can't get close to them with
Yeah, but have you tried to using the outputs from the other servers? Then we have our outputs. I mean, taking the
That's all we have access to.
Okay.
You know, we send in what's on your supplemental materials. Here's the test set.
Yeah.
So we take the test set, throw it through your website, get the predictions back.
Yeah.
And then compare your predictions with our predictions.
Oh yeah, but okay.
Oh
yeah, but then again, but also not only in the integration, but also on the MHC predictions, we also have access to a huge number of data that are not published. I mean, all the boost data, they will only come in later versions of the IDB. So
that would explain.
and that really means a lot, of course.
Yeah, there's no data like
More data, that's our...
No, not in this. In this is, I mean,
So for right now in the data that you can train on, which is not publicly available, not in the IDB. So like, what would be the number? Like there are 35 alleles in IDB, I think. 35 HLA class 1 alleles. So on those 35 alleles, just to get a feel of it.
Okay,
like,
then
what's the size of the data that you have, like for the smallest allele? Like, right, AO201 has like 3,000.
I think but for I think we would. I mean, the one that we will not train network on something that we have less than 50 data points on. So I mean, because in that case you can get a decent matrix out of it, and then maybe the network will only learn the matrix, but that will be fine then anyway. So let's just. Yeah, of course we have the extra matrix data from the Suffici. So that counts.
The data is again allele specific, right?
Yeah. So it's hard to say how many data you really have in combined. So but at least 50.
Data is only used for that additional feature.
Yeah, that's but if you have very few of real binding data, that is, I mean, this additional feature is very important, because I mean, it will gain a lot. The network will learn, okay, if I just take to trust these 9 inputs, then I will be right in most, and I can't figure anything out of just using the other inputs. So
why don't you use the safe pathing data as part of the training for the null network directly? Because it's not suited for that.
Yeah, it's not suited because it's not. You don't have an affinity.
Oh, you only have a binary.
Yeah, yeah. But we should maybe look into making the. No, we don't have. Yeah, no, but it's.
If you make the network binary, then you just have a lot of data.
Yeah, but that's not as good. Then you don't get a very. We like to have a real affinity out of it. Otherwise, you would have to afterwards go in and make some adjustment to see to get your to get an output score corresponding to some affinity. As it is now, the networks should be learning it directly, so the output from the network should be the affinity that the network is predicting. So we don't have to.
But if you try to optimize binaries, like binary prediction error, if you were to do that, could you get better scores if your output was binary? So like in IDB, right, they have the IC50 values, but then
that's also
everything below this and this is a binary or a non-binary, and everything above, or the other way around. I'm always confused. But if you actually threshold the data, would you get a better results if you trained a binary neural network? On all of the data?
That you mean on then the AUC results?
Yeah.
I mean, you would probably, I mean, it depends on what you train it to. I mean, if you really train it to get over and below or below some special threshold, and you know that threshold, then you probably would get better to that definite threshold. But it is actually nice to get the affinities, because I mean the threshold you usually set is like 500 nanomolar. Above that it's a binder, and below that it's not a binder. But there is a lot of fine-tuning that says that sometimes an epitope can go to be 2 micromolar, really, it seems. In some cases, one of the where we have some influenza data where we have tried to where we get on compromise, where we didn't were so hard on the binding threshold, and we saw that sometimes that really an epitope is only.
Can you explain that by these other effects? When you add the other effects like cleavage and tap and whatnot?
Yeah. Probably it's like, I mean, they have very good. It could be that they have very good cleavage and very good tap affinity. Or maybe it's just that they are expressed very high. I mean, they're highly expressed proteins, so they would have a good chance of getting out there anyway. Or it could be, I mean, we are predicting affinity, which is equilibrium. It might be that they are very stable, even though the equilibrium is slightly high. There might be that it's because the on rate is slow, is fast, but the off rate might be that the on rate, it might be have a high. I mean, have a weak affinity because the on rate is slow, but the off rate is also slow. Maybe, and even slower. So it might be that even though it's not have a very good equilibrium, it might be that when it binds, it's very stable, and then it can stay there and still make so there's all these things. So in that case, it's very nice to know the real affinity and not just having a threshold. Another thing is that it seems also for different diseases we're trying to make cutoff, and it seems like for example for influenza. It's like using a 50 nanomolar cutoff is, I mean, if you're just going for being very sure that your predictions are epitope, that is a very good cutoff there. But for some other thing, for pox, where we have a very old, this is vaccinated people, so they have been vaccinated over 30 years ago, very old memory, we have to go down to 5 nanomolar. To be able to have, I mean, we have like, but I mean the statistics there is still weak, but we have like, I think 11 verified epitopes from pox, and they are all below 5 nanomolar, and nothing between 5 and so for these reasons it's actually very nice to have the affinity, even though you might be able to having some better Matthews correlation about some specific cutoff.
So you're saying that it may be the case that for different diseases,
the cutoff that determines.
No, it might also be the age of the memory cell, because I mean there's probably the number of the T cells decreasing all the time. So be able to trap one rare thing, it has to have a stronger binding to be there a longer time, or whatever. What do I know? And the influenza one is more recent, because we have also just taken donors. So here we have taking healthy blood donors between 35 and 65. So, and these are expected to have had influenza more than 3 times in such on average, or a good chance. So, but it's probably not 30 years ago since they had the influenza. It's probably maybe 10 years ago or 7 years ago then since they had it the last time. So it's a newer instance memory. It's a newer memory. So, and that might be the reason that you have that.
It's only 15. I mean, usually people use a kind of 500.
Yeah, but that's because what you see on the eluded. I mean, if you're taking the Cepheid data and you actually measure them, some of them will also be like 2 micromolar, but you have a strong drop in the number of peptides you see that are weaker than 500. So the 500 nanomolar is what you see when you elute them, but that are peptides that are coming all the time. So that might not be strong enough to generate a real memory. It might be strong enough to generate a response, but not to generate some long-lasting memory.
500 nanomolar. Which way? I'm confused. So you mean the
one below between 550 nanomolar might not be strong enough to create a long-lasting memory.
Why?
Because I mean but it still have to what?
Is it the same idea?
It binds quickly, but it comes out of the groove quickly as well?
No, I think it's something that there will be less of them. The number of the MHCs that actually have an allele bound would be less. So the chance that a T cell receptor would actually bind to them would also be less. So you have
presented less often, or
why would they have a higher affinity?
I'd rather use the term weaker or stronger, because that is because
Weaker affinity.
if they have a weaker affinity,
Then I understand, but if they have stronger affinity,
Oh, they're
it's
weaker.
5 nanomolar.
if they have a stronger affinity, then you would have a higher number of memory cells in the end, because you get a
500 is weaker. I mean, 500 is the threshold.
yeah. So maybe 500 is not a good threshold for epitope, but I wouldn't say that it is in some cases.
Memory dies away. You mentioned that memory maybe dies away, and that's why for all infections you need very strong binders, right?
Yeah.
So how quickly do you think this memory fades away?
I would know. I'm not a real immunologist, so I wouldn't know, and I wouldn't bet on it. But it seems that, out of these two cases, it seems if you're going there, you see you have after 7 years on average, you can have a response of 50 nanomolar, and after 30 years, you have to go up to 5 nanomolar. Go down to 5 nanomolar. So but if it's linear, if it's something like go like this or whatever, I wouldn't know. So it's and I don't think that anybody knows. And it also might be pathogen specific, and also again how expressed is the protein that's generating the response in the first place. How much is coming out on the surface of the cell.
So it may be that things are not very good binders, but just because they're expressed a lot, then they get presented a lot, and then they cause a reaction.
Yeah, and that and if it and if it does that, but it doesn't seem to last long anyway, even though it's low. The ones that we see.
They don't seem to have a long memory.
Long memory, yeah.
Even though they may cause.
But you couldn't say that. It also might be some competition issue here. It's too few data to make this this conclusion on. So yeah, it's ELISpot that has been used to indicate it. So here we have, this is for the influenza. So you see, even though we have quite a high high number of binders, it's only like then 10% of the binders or something like that would be actually epitopes in that number of donors that we have tested, of course. But the interesting things about these epitopes is that they have been selected on being very conserved in H1N1 influenza, and then actually many of them are also conserved in the bird flu. So these might be interesting epitopes in the long term.
So you mean that the ones that were discovered so you had a bunch of binders.
Yeah, okay, I should.
Them for epitopes.
Yeah, this
you
is.
only found that 10% were epitopes.
Yeah, actually, not only 10% of the binders were epitopes, because we wanted this very high number of high level of conservation between all the strains of influenza. We didn't look very hard on the binding threshold. So some of them was really predicted not to bind, but we wanted to
that were highly conserved. You took areas that were highly conserved.
Yeah.
and then tested those for binding and for
They were also predicted. I mean, they had we also ranked them to the combined score for being epitopes, but the threshold we put a little lower than we all otherwise would. But we wanted to see where does the threshold have to be also. So so half of them were binders, really, when it came to it.
To Imate C.
To Imate C, yeah. And 10% of the binders were then actually epitopes.
But the epitopes were more conserved than the others, or
No, everything was conserved. So everything was selected to be conserved.
Conserved regions selected 180 peptides to test, and they tested both ways, as binders and as
Yeah, and we did it the other way around. We predicted a lot of epitopes, and then we took the ones that occurred most times in the strains. Yeah.
okay. So you didn't start from a conservation
score. You started from predicting epitopes.
We started from predicting epitopes, but we predicted everything.
Okay.
We have prediction
on the entire strain.
on
all of the strains.
Yeah.
Okay, and then you look for things that popped up in many strains.
Yes. And then what actually determined the combined score threshold was this number. We wanted this number of predictions.
Okay. And if you would have gone the other way around, what's your intuition like?
If you would have looked for conserved regions in influenza and took
I think you would ended up having, yeah, but a little bit there. Yeah. Yeah, probably. Yeah, if you're looking for conserved 9 MERS.
One way or the other, they limited themselves to look at the conserved 9 MERS.
But anyway, it wouldn't be exactly the same because for the epitope prediction, you need the context also. So the context might be different for 2 strains that have the same 9 MER. So these might not popping up in all of them as in the same range. So it's
Did you only test the binders to see whether they were epitopes? Or
no, we tested everything. So
non-binders that were epitopes?
Yeah, a few. That 1 or 2, I can't remember.
It's the T cell receptor that's stabilizing the binding.
We don't know, and it might be the T cell receptor, or it might be, as I said, how stable the complex is when it first actually have bound the peptide. And it would be very nice to actually look at this biochemically to see what does it mean. And I think it means something because actually I haven't shown the BMAS predictions. That is half-lives. So that is actually how stable is and they're actually pretty surprisingly good as predicting epitopes. And we have never done that, but I would like to actually combine it to our method also to see for the ones where there is BMAS matrices if they can actually improve, and that would indicate that half-life is important also for this thing. I think it might be.
So this data set is one of the
first things I've seen that can help you differentiate binding from epitope, which is an important thing to do.
Yeah.
I mean, is that kind of the key goal here, is to focus in on this difference? The difference in prediction?
That is something we want to get out of it. The key goal here is to identify epitopes. That's what NIH paying us for doing. So it's
right, but you've got these. You have binding. You have
yeah. So
the T cell receptor or whatever.
So it's very nice from this that we can see how important is the different steps. But that's what I say. In this, I don't know if I have not. But in this case, what comes out of it is that actually binding is very important, and that I think 80% or something of the epitopes here was below 50 nanomolar when you measured them. So that indicates that actually it. So. Yeah. If you really want it, you should use it. But if you only want to be quite sure to have some immunogenic peptides, then you should use your 50 nanomolar cutoffs, and your workload would be much less for a high amount of discovered epitopes.
So have you found any patterns that predict why there's a difference here?
Why there's a difference between
binding and CTL?
No, not that we have discovered yet. We would like to go into we would like to maybe soon our PhD student is looking at this thing about how close the epitopes are to self, for example. That might be something.
Back to the question before. You did have some examples of things you thought would be good binders, so you tested them, but they turned out not to be good binders. And nevertheless, they turned out also to be epitopes.
No, that's what I say. We did not necessarily have predicted these to be good binders. There was we just ranked them to a combination score, and this number determined the threshold. So some in the bottom.
Combination score, yeah. So
No, but he was saying that they
combination
just...
score. In the combination score, did you have some of these binders, some of these peptides had a combination score that takes into account cleavage and tap and everything that was good, but they were not good
But
binders.
he's saying they're
according to
not
prediction.
all good because
They're
they
not
selected the number
necessarily. They're not necessarily all good. Some of them. I mean.
They must be. I mean, it's only 180 peptides. You must have found 180 good peptides.
No, because you have the influenza are very, actually very diverse. Even if you're taking in 1H1 strain.
Threshold on 70%
conservation.
Yeah.
You may end up with not a lot of peptides to begin with.
So that's the.
With some things that you predicted would not be.
Actually, if to our standard threshold, we wouldn't have taken them into considerations because
By the way, did you compute the correlation between the conservation and binding?
No, but yeah, you could say because.
It would just be interesting to see whether things that are more conserved bind better.
Yeah, worse.
Or worse?
Yeah, because that's why they're conserved.
Sure.
I mean, otherwise the virus would have get rid of this.
That's a good question.
So,
I don't know.
So then you had a prediction for some of these things that are bad binders, and then your predictions turned out to be correct, that they're bad binders?
Yes, there's.
But back to the question. So you had some things that turned out to be bad binders in the experiments, but good epitopes.
Yeah.
And how many of those do you have? Is it a handful or?
No, I think there's only two epitopes that were actually weaker binders than 500 nanomolar. And 80%.
Was your score for those combined score was it?
Well, that was lower than our ordinary threshold we would use. But the binding is the major part of the threshold. I skipped that, but it's like we're putting. Maybe I didn't
weights
even.
of the mixing. Yeah, sure.
I forgot to have the weights, but the weight is. I mean, we have putting 1 on the binding and 0.15 on the tap, and then 0.05 on the proteasome cleavage. So the binding is far the highest part of it. So.
Can you stick on that slide for a second?
This one? Yeah.
You said you've got a paper in submission on this?
Yeah, we have it right now. It's we have it just get it turned down, so we have to find out what. But this is a strange thing.
A request.
Yeah.
If these results if any results that you have in the paper are based on training on peptides that you're not allowed to share.
Yeah, they are.
It would be great for the whole community if you can leave those results in, but if you could put in new results that say on peptides that I'm allowed to share with you, and here are the peptides. Here's the results you get.
Okay.
So that we can compare.
That's a good idea. Yeah, but actually. Yeah, but I think the evaluation. Yeah, okay. It's impossible to. Yeah, I see what you mean. It's impossible to come along and say. On the method, on the basis of the method, it's hard to compare. Yeah, you can compare the final version. The data is but it's I will.
It's extra work, but it makes it really great.
Yeah, but I think that might also get it because there's been a lot of suspicions. We have. The reviewers has been very suspicious about this. So. No, it's okay. I don't think so because one of the things we got from reviewers was some things that you actually also did. I mean, using that was for binding, using just random peptides as negative data because and one of the reviewers said you could not do that. I don't know. I mean, it was no, but let's
as opposed to established negatives.
Established negative, there's a huge bias in the literature. It's
but
someone chooses to establish as a negative epitope could have all sorts of confounding variables.
Because that was an epitope, but I think the reviewer misunderstood it and thought it was binders. So anyway, but even on binders you can do it. We do it because we are boosting because if we only have 50 peptides with data on, usually many of these are binding peptides.
Sure.
So we boost our prediction and say we're taking 100 randomly selected from Swissprot peptides and say these are non-binders.
Right, sure. But if you look at the distribution of non-binders that's in IETB,
it's very different than random. It's biased, but it's not necessarily biased by the experiments. It's biased by the data because usually these non-binders are things that were thought to be binders.
Right. So you actually have a signal. That was
they're really hard. That they're very hard to distinguish from binders.
But okay. So in that way you can say that's good. So they have high value.
Yeah. You can actually show that if you just randomly
initiate a PSSM to distinguishing non-binders. If you just build a PSSM that just looks at a very simple signal, you can distinguish. You can throw away most of the random non-binders at ease. And even if you don't have any neat learning method of any sort.
But
there is a big difference between these.
But on the other hand, you can also say that
if you don't have a lot of real non-binders, then you should use random.
Yeah, but also it's what you say is correct, but it's also usually they have been selected to bind to something, and then but it might be that they are binder to something else. And actually, in what we had, all these boost data, all our negative binders were something that, for 90% of them, was something that then bound to something else. So you got a bias, so you can get a network that can distinguish, can it bind to this or can it bind to that? But not all the things, they haven't learned all the other things about things that don't bind to anything at all.
Sure. When you have a non-binder that's experimentally determined, you usually know that it doesn't bind to something specific. It doesn't mean that it doesn't bind to everything. But all I'm saying is that this is where it's really hard to distinguish, right? Because if you just take a random non-binder, if you just take a random peptide from Swissprot, it's usually if you take a human immunologist and say, is this going to bind? You can just look at it and say no. And he's going to be right 90% of the time, 95% of the time, because he can just tell by looking at the peptide.
You don't even have to look at it.
No, you can just say no. You can just say,
Sure.
But
I'm saying, you're right.
I can say that there will be. I mean, as when Booz is finally releasing all of his data, there'll be a huge number of non-binders, measured non-binders.
And when will that be?
What?
When will that be?
I don't know. I mean, he has to do it at some time because that's
part of the grant.
Yeah, that part of the grant. So,
but do you know if it's going to be like this year or like in 5 years?
No, I think he had another grant from the NIH where it has to measure 50,000 data points, and a lot of these will be. I think they're already submitted. So I think they are in the pipeline right now. Within.
So they should be going out.
Yeah. And there's a lot of non-binders because he has this thing. So whenever he has the peptide is the expensive part. So the measurements is just because he has this robot and stuff doing it all. So it's not just the measurements are not expensive for him in labor work. So he can measure on all his panel of whatever he has 40 MHCs the same peptide. So you get 40 data points for each peptide. And most of them will be non-binders, of course.
Can you do like when you do these binding assays, right? You always take a peptide and you put it in a you take like a bunch of peptides and then you put them on the same MHC. But can you do it the other way around? You like put a peptide and put a bunch of MHCs, and then figure out which one of them is going to bind.
Yeah, but that's basically what he do.
At least what is like this reaction.
No. But what Booz is doing is biochemical binding. So that's not ELISpot.
I know.
So he's just, and it's in ELISA plates. So whenever he's having the same peptide and all his and different MHCs, or you have the same MHC or different peptide, it's just, and I don't know which way around he does it really. I think, actually, he does. I don't know. and it doesn't really matter. But he's doing both.
Once you get the peptide, you can test it on all the MHCs. For
Yes,
the same,
yes.
so
All
this
his
is right.
peptides will be tested on all his MHCs.
Oh, that's very interesting.
So that's very nice also. So we get a lot of negative data that way also. But it's still nice. I mean, it still helps the networks to boost them with real. I mean, with the ones that has never been selected to anything else. Yes, I should go on with this. And that's the final thing. That is if you want to. Now this was the conservation. So this one, all the peptides has been selected to be in as many as possible as of different strains. But maybe you want to have, if you have 180 peptides, you want them to cover as many strains as possible. And that's a different question. So we have tried this schedule out where then they all have to be binders, have to have be above the combined threshold score. So select peptides of the above threshold score with the highest that covers the most of the genotypes. Then in the next round, you're taking the next peptide you select is the one that covers most of the ones that are not selected by the first peptide. So in that way, you are covering most of the possible genotypes. And yeah, this is just a test with some HCV data, and that worked fine, and we got they're predicted to bind, and these are how they covered. And then from just this 5, you could go in and look in the Los Alamos HCV epitope database and find that one of them is actually there. But we're using this approach on HIV epitopes. Okay, where did that go? Okay, I don't have a slide on that. Oh, there. But we have used that approach on HIV epitopes. We have selected again around 180 peptides, I think, and they have been tested in pools. Annika Karlström in Sweden have tested these peptides on 32 patients with HIV, and found that 31 of these 32 patients actually reacted to one of these 180 peptides, and that 66% of the peptides had a reaction in 180. So that will be
roughly 120.
But
120.
We've tested related peptides on a bunch of patients, and we got that always a patient will react to at least one of the peptides.
Yep. Yeah, but you have select
larger number of peptides.
just as a random peptide, right? A random peptide?
They're not. They're just
Taken from a genome, right? But
Yeah, no. But I mean, they have not been selected to be epitopes at all.
No.
we tested everything.
We just tested everything.
Yeah, and
If
every patient is going to recognize something.
Oh yes, but how much do it? Okay, if you're just taking 100 or what, or how many do you take?
We took about 200 peptides maybe.
Okay, so if you're taking 200 peptides,
944.
Peptides.
Right, but they're all related though, right?
Yeah.
I mean,
What's the question?
the true number of peptides that we really have different
From the
peptides.
consensus strain, if you think.
I mean, the question is, given 200 peptides taken from consensus or from anywhere,
Okay.
what's the chance that a patient is going to recognize something? Chances are very high.
Yeah, about
Everybody's going to
95%
recognize.
of the 18mers that Nicole tests.
Yeah, they all get hit.
okay.
Yeah, 25% get recognized
From
by somebody from her pool. But if you look at a patient, the chance that the patient is going to recognize something on a 200 long sequence is extremely high.
But in this case, okay, but then in 66% of these peptides was recognized by somebody in the pool.
Well, 66 of these peptides.
Peptides was
No,
recognized.
66%. So that's 120 peptides.
And these are 9mers or whatever.
These are NIMES.
So these NIMERs are, okay, so most of your NIMERs are recognized by all patients, is that what you're saying?
No.
No. There's different patients recognize different NIMES.
But that's what I'm saying is not highly unusual. That's what.
Yeah, but you said you had. It's 25%.
And without selection.
You said 25%.
You said 25.
But 25% is that an 18mer is going to recognize.
Yeah, we have 66% of these NIMES. So you are
60% of these dimers is
I'm really
recognized.
confused now, I don't know.
so it's just
Experiments, for example, I don't think there is a single. I mean, maybe there's a few temers that are not recognized by anybody. I mean, almost every peptide is recognized by somebody. In NEF, anyways.
But you say you're using 18 MERS.
No, that was a different test. I'm talking about 10.
10 MERS. And 10 MERS.
You have to describe the experiment so you can understand what you're talking about.
But anyway, if you say let's.
Tested all 10 MERS from that.
Yeah.
So they took all 10 MERS from NEF and they tested like on them on a bunch of how many patients?
30 patients.
30 patients.
Yeah, it's the same model then. Yeah. And
even though they're not selected to be epitopes, most of the peptides are recognized by one patient at least.
Yeah, okay, but that's the ones. So I say that part of it might not be significant then it's not. But the other number is that you actually need to investigate a far lower number again of peptides to getting the same number of epitopes out of it. Because or actually you're getting, I mean, because 66% of the peptides here of the 9 MERS was actually recognized in at least one patient.
You think that in NEF?
Every peptide gets recognized by...
66, I got to check, but I would bet at least 66% of all peptides in NEF are recognized by at least one patient out of 30.
Well, we have to check that.
I don't
tell you.
Higher, or you can tell us in a minute? Okay,
Okay, but that would be interesting if it's so. Okay. Because then you could say this is not different from random then. But then again, it would be hard to get over random.
Huh? Over?
Yeah, better than random. If anything is recognized in somebody, then it's.
But you could start looking at if your
Peptides are recognized. The number of peptides you have predicted that are recognized in 5 patients out of 30 is unusually high.
There's some of the peptides are recognized in 4 patients.
So maybe that number is very
But
good.
yeah, but you will see Annika Karlström will have a poster at Keystone here with the numbers on. So.
What day?
What?
What day? What day?
Tuesday, Wednesday, Thursday.
Oh, same Tuesday, I think. But I can tell you because it's the same day as we have our poster, so it's. Then it's class 2 here, very fast if you do. We're using something called a Gibbs sampling approach. Where we have a Monte Carlo simulation that tries to move around a 9-mer core until you have the highest signal within the alignment. So, you move the alignment around. So the final alignment have the highest possible signal in it here.
So use the Monte Carlo
You use the Gibbs sampler to align the data.
To align first.
To align the data,
Then
yeah.
once you have the alignment, then you use it as
Then I use the PSSM that was total. That's what we're doing now. Yeah, and yeah, and you just see that if you're just using like something like Tepitope or ProPred, it's the same. Within these without aligning them, just within this to select the best epitope. You won't get as good a score as if you.
What's Tepitope?
Tepitope is the one that Jürgen Hammer. That's the ProPred is really an implementation of Tepitope. So Jürgen Hammer made this from peptide libraries. He used from phage display, used scanning to see the 9-mer cores that were binding to different class 2 MHCs, and then he used that you could interchange the pockets. So in that way he can predict on a higher number of different alleles, because some allele has the same pocket. There's this 3 major pockets in the class 2. Yeah. So and that's, I mean, this is the one that people still compares to, because that is.
So who did that?
Jürgen Hammer. Originally, now it's Ragava has made this implementation that is free, because his program was not it was something that was only working on PCs, and you could only get his program. So, but the data he had used for the matrices was actually, or the matrices itself even, was actually published. So this guy Regava, he went into the literature and took out the matrices and put them on his website and used a mega web implementation of using the same matrices. So you can go in there. And actually you can just take what we have done, our test of it is actually taking the matrices and just using them in-house. It's much faster, of course.
That third. Of the whole
44, right? Not the consensus ones.
Well, of the 944, which aren't just consensus, one just consensus.
Yeah, what I was
944
looking at
consensus.
per position, which positions are recognized, which are not. Well, out of the 206, and most of them are recognized.
Oh, out of positions on.
Right.
Oh, I don't know.
Because the idea was that if you just
Because some of the things that
take
we are
everything
testing
and-
are very rare, so we're not expecting reactions anyways.
Oh,
Just look at the consensus of the markers.
so yeah, I don't know. We have made a cell lab now and also doing some 2D proteomics for B-cell epitope discovery, antigen discovery.
So what are these 2D gels?
Yeah, that's for, we want to do antigen discovery for malaria and TB.
What sort of antigen again?
This is just on the proteome. We have just mashing the parasite and doing and see. In the first case, we're trying with established antibodies to see if we can recognize the right antigen. It's in the very beginning. If we can recognize the right, and later on we will use like polyclonal antibodies to recognize some important antigens in this way. So.
You just want to search for things that are highly antigenic.
Yeah, and then of course in the end we would like to be able to find out which epitopes in the antigen are responsible.
So what's the 2D gel technology? What is it? What does it do?
The 2D gel is just a way to spread out all the proteins that are in an organism.
And you can identify
the areas which are antigenic. How do you do that?
You can do it with antibodies. You stain them just like.
antibody and just look at where you're going to stain.
Yeah, look where it's going to stain, and then you can plug out that from the gel actually and put it to a mass spec and then get an identification of what kind of protein are. Maybe if you're lucky and there's not too many different proteins in the sample stuff. But yeah. And we are hoping to go on with some mouse immunizations against TB also in-house right now. Yes. Yeah. We are going, as was said, going towards systems biology. And this is a page of our course, but, if you want to stay up at night. So yeah, that was it. Took a little more than an hour, I'm sorry.
We ask questions. Thank you.